For those of you who don't know me, I'm Arno Kumigai, Vice Chair of Education for the Department of Medicine, and I have the special privilege of introducing this year's Levinson Lecture. This is a special lecture that was created in 2015 to recognize a senior member of our department for their outstanding career contributions in medicine, research, education, leadership, or any extraordinary contributions to patient care. And as you recall, in the past, this is obviously named after the former chair, Wendy Levinson, who's on this call. We've had speakers in the past, including Ian Tannick, Jay Keystone, Michael Hutchin, and most recently, Sacha Bhatia for quality improvement. So this year, I have the absolute pleasure of naming the chair on behalf of the Department of Medicine and Jillian Hawker, our current chair, naming the Levinson Lecture for Tony Lang. Tony has a whole alphabet of letters after his last name. He's incredibly well recognized. He is a full professor, previous division director for the Division of Neurology here at the U of T. He holds the Jack Clark Chair for Parkinson's Disease Research. He is also the director of the Edmond J. Safra program in Parkinson's disease and the Morton and Gloria Shulman Movement Disorders Clinic, and also holds the Lily Safra Chair in Movement Disorders at Toronto Western Hospital. He's published over 700 peer-reviewed papers and was the most highly cited investigators in the field of movement disorders. Among the many awards that Tony has received, he was appointed the Officer of the Order of Canada in 2010. In 2011, he was appointed a Fellow of both the Canadian Academy of Health Sciences and the Royal Society of Canada. In 2014, he was elected by the International Parkinson and Movement Disorder Society as an honorary member, quote, in recognition of his extraordinary contribution to the field of movement disorders. In 2017, he was the recipient of the first MDS, that is this Movement Disorder Society Pan-American Section Leadership Award. And in 2018, he received the Western Brain Institute International Outstanding Achievement Award for work in accelerating the development of therapeutics for neurodegenerative diseases of aging. So it is with great pleasure I am passing the mic over to Tony. So Tony, congratulations and welcome.
Thank you, Arno. It's a tremendous pleasure to have been asked to give this lecture. We often say when we get introduced that way, thank you for that kind introduction, but I think everybody on the call needs to recognize that we usually provide all that information to the people that introduce us. So it's a bit built up. So let's move on if we can get, okay. So my topic, as you can see here today, is to deal with neuroprotection in Parkinson's disease and to address whether a single drug will ever do this or whether we're really going to need to think beyond single drug approaches. As has been emphasized, this is the Wendy Levinson Lecture. Tremendous pleasure to have been asked to give this, a tremendous honor and pleasure. Wendy was my chair when I was the division director for most of my time during that period. I stayed on a couple of years afterwards, and I learned just so much working with Wendy. It was a joy, and I really appreciate all of the novel information that she provided in administration and development of quality of care, et cetera, in her role as the Sir John and Lady Eaton Chair. So I have a number of disclosures. This is a very exciting time in the field of Parkinson's disease, and I'm very pleased to be involved in drug development and advising companies and researchers on this field. It's a frustrating time and a frustrating field as well. My objectives are listed here. We're going to talk about what we mean by one drug or combination therapy, justifying this discussion, emphasizing the heterogeneity of the disease, and questioning whether we're dealing with a single disease or possibly multiple diseases. We'll then talk about the reasons that we might need to approach this field with multiple drugs in terms of the concepts of drug development and clinical trials, as well as disease pathogenesis, and then we'll conclude. So this is a slide that I often show of all of the tombstones in my field of neuroprotective therapies. I was involved in many of these trials. You see the drug names on the left, a variety of different agents studied because they had very good preclinical and some clinical evidence to support their use with many different mechanisms of action, using many different trial designs and different outcomes. And unfortunately, despite thousands of patients participating and millions of dollars invested, none of these were effective or successful. That doesn't mean we've stopped, and this area is still very, very active. And this is a list of various targets. I'm going to cover a little bit of this, but we're not going to get into great details. Many different mechanisms of action, a large number of drugs. And on this slide are only those drugs that are in phase 1, 2, and just very early phase 3 kinds of trials. So this doesn't even cover all the drugs in preclinical development with all the different mechanisms that have been proposed. Could we get maybe people to mute their mics, because I'm hearing a little bit of feedback. Thanks very much. So the question of one drug versus combination of drugs. Well, what could we use as definitions here? Well, one drug could be a single drug for everyone. Or does it mean that you would use a single drug, but it would need to be combined with other drugs in various combinations, depending on the biological subtypes of the disease you're interested in? Or multiple different drugs targeting different pathways or different pathogenic mechanisms that I'm going to introduce you to, or multiple different components of a single pathogenic pathway, again, that we'll describe in a bit. So why might one drug not actually fit all patients with Parkinson's disease? Well, this is an amazingly heterogeneous disorder, and I'm putting PD in quotation marks, and I'll explain that in a bit. And the heterogeneity is seen in all of these different areas, and I'm just going to quickly touch on each of them to introduce you to the complexity of what I believe is the most complicated disease in medicine. I really do believe that this is a very complicated clinical, pathological, genetic, environmental, etc. disorder. So does heterogeneity imply multiple diseases? Well, I think it's pretty obvious in any field that it does not. We could have different clinical phenotypes of the same or presumed same underlying etiology or pathogenesis. And a very good example of this is monogenetic diseases. At least in neurology, we deal with a large number of monogenetic diseases that can manifest in a very polymorphic or heterogeneous fashion. Huntington's disease, for example, in a child looks entirely different from Huntington's disease presenting in an adult. Different clinical and pathological phenotypes with the same etiology, question mark pathogenesis, I say here. And you can think of many different conditions. I'd give a couple in neurology: tertiary syphilis with general paresis of the insane versus tabes dorsalis. And prion diseases, Creutzfeldt-Jakob disease, with many different clinical phenotypes that look entirely differently when we examine them and assess them. So let's quickly look at clinical heterogeneity in Parkinson's disease. This is a slide that just shows you multiple attempts from the late 1990s to close to now, multiple attempts using data-driven approaches to subdivide Parkinson's. And you can scan this. We're not going through it in any detail, but for example, patients have been divided based on the speed of progression, the age of onset, based on clinical features, whether they have a tremor or not, and various other different methods of separating patients on the basis of their clinical features. None of these, however, have been reliably reproducible, and none of them have been used to subsequently study disease-modifying therapies in the clinic. This is a study of the pathology of patients with Parkinson's, subdividing the clinical features under mild motor predominant, an intermediate form, and diffuse malignant. You would have expected that this might have been represented, these clinical phenotypes might have been represented by different pathological types. The investigators found that these clinical features were useful in estimating disease course and survival, not surprisingly. But when they looked at the pathology, they couldn't stage the severity of the Parkinson pathology or concomitant pathologies in any way that correlated with the clinical features. So let's look at pathological heterogeneity then. You probably know, I hope, but if you don't, this is very important. All neurodegenerative diseases now are subdivided based on specific proteins that deposit and aggregate and that we believe contribute to the pathogenesis. For example, in Alzheimer's disease, the proteins are a-Beta and tau. In Parkinson's disease, the protein is alpha-synuclein, a normal synaptic protein with a number of normal functions. But we know now through initially genetic studies of a large family that originated first in Greece and then in Italy that immigrated to the United States, were found to have this first form of alpha-synuclein mutation. We now know of several other mutations and other genetic variations, all predisposed to the development of Parkinson's. And once the A53 mutation was discovered, Spilantini used immunohistochemistry to show that the Lewy body that many of you might remember in your studies early on, this inclusion body that is a hallmark of Parkinson's, she showed that the main protein that was the constituent of a Lewy body was alpha-synuclein. And staining for alpha-synuclein showed an extensive amount of pathology we were never aware of before this staining. And this shows the so-called Lewy neurites associated with Lewy bodies. So we now talk about Lewy pathology. There is also a belief now that we can recognize the presence of this pathology that, based on work by Heiko Brock, that the pathology seems to, in many cases, maybe most cases, spread from the low brainstem up through brainstem and then subsequently across the cortex. So he introduced the staging approach to Parkinson's pathology, indicating that synuclein pathology spread in a way that some people believe is a cell-to-cell transmission kind of process. So, synuclein and pathological heterogeneity. Well, despite what I've told you, we now know that there are many exceptions to this model of spread through the brainstem and up to the cortex. Some people seem to have, on postmortem, disease in the cortex primarily, or in the high brainstem without low brainstem involvement. There's some belief that the disease may even begin outside the brain, in the enteric nervous system, for example, in the gut. And there have been recent studies, we took up one in our journal club this morning, of the concept of some patients with brain first and other patients with body first subtypes of Parkinson's disease, dependent on where the synuclein pathology originates. And then we also know that you don't even need synuclein aggregation to develop the neurodegeneration that is part of Parkinson's disease. If you look at patients with the earliest stages, the BRAC stages, where the substantia nigra, the dopaminergic cells, are housed, we can see loss of nigral cells even in the absence of Lewy pathology in that region. Another really fascinating finding is this genetic form of Parkinson's, LRRK2, the commonest genetic autosomal dominant form of Parkinson's disease now. And I'll show you some pictures that originate from these patients. And then finally, we now recognize that it's not just the synuclein pathology that causes Parkinson's. Co-pathologies seem to be the rule. This is a fascinating study from a single family with this mutation in the LRRK2 gene. Some patients with classical Lewy body brainstem pathology, others with additional or even distinct cortical Lewy body pathology, but others still without these features who have neurofibrillary tangles, a progressive supranuclear palsy-like pathology in the brainstem, and still others. And the first brain of one of our families with LRRK2 that came to pathology had this, a bland neurodegenerative disease without any synuclein seen despite extensive attempts to define its presence. So a bland degeneration with loss of nigral dopaminergic neurons without synuclein. Then there's this co-pathology issue. This is an important study that looked at patients who had synucleinopathies, Parkinson's disease dementia, or dementia with Lewy bodies, synucleinopathies manifesting dementia. 77% of these individuals had additional Alzheimer pathology. This is work from the Mayo Clinic with 140 cases of Parkinson's disease, just showing you the frequency of other co-pathologies. They found not all of these patients had dementia, so here about 40% had Alzheimer's, but some had PSP and other co-pathologies. And this is work from Virginia Lee and John Trojanowski from the University of Pennsylvania, looking at a large series of patients with Parkinson pathology. And depending on the stage or the severity and distribution of the Lewy body pathology, they all had co-pathologies, and some, for example, where the limbic area or the cortex was involved, there was much more extensive protein deposition and multiple different proteins were involved. And the more those multiple proteins were present, for example, Alzheimer pathology, the faster the cognitive changes developed and progressed in these individuals. Okay, so now let's move to another level of heterogeneity: genetic, environmental, and pathogenic heterogeneity. We now recognize that Parkinson's disease is a genetically heterogeneous disease. I mentioned LRRK2 is the commonest autosomal dominant form of Parkinson's. Interestingly, GBA, the gene for Gaucher's disease, glucocerebrosidase, is the commonest genetic risk factor. We would have never believed that until the last several years, where patients with Gaucher's disease had relatives who were heterozygote carriers of the gene were found to have developed Parkinson's disease. But this slide also shows you a large number of what have been called the PARCs, demonstrating that there are a number of monogenetic forms of Parkinsonism with many different genetic underlying causes. But distinct from that, doing studies, for example, with genome-wide association studies, we now recognize, this is the most recent report in a year ago by Nals and his colleagues, 90 independent significant genetic signals accounting for between 16 and 36% of hereditable risk. And we're now learning that if you calculate a genetic Parkinson risk score, you can actually predict the risk of developing Parkinson's disease. And there is a move afoot to use these genetic risk scores in planning clinical trials. For example, studying patients with a higher genetic risk score with different types of drugs that might have a greater impact on the progression of the disease. In addition to using those genes that increase risk, there are also genes that reduce risk. And there are a multitude of environmental risk factors that both increase and decrease risk. We're not going to go through the details, but just to recognize how complex this is a multifactorial disease, and there is a clear recognition of the need to look at both the presence of these factors and also their interactions. And this is, for example, one of the earlier studies that looked at an HLA amino acid motif that predicted and accounted for the role of smoking in Parkinson's. Many of you may be aware that smoking reduces the risk of Parkinson's disease. The mechanism's not known whatsoever, but it does seem that there may be important genetic factors that contribute to this. What about pathogenic mechanisms that show the heterogeneity of this disorder? Well, you can scan this slide to see that the many different cellular pathways are believed to be involved in Parkinson's, either independently or in concert to contribute to Parkinson's, from clearance systems that get rid of aberrant proteins and aberrant mitochondrial, through to homeostatic mechanisms of mitochondria, calcium homeostasis, endoplasmic reticulum stress, and particularly in all neurodegenerative diseases now, the role of both the innate and the general inflammatory system. And so these factors can occur both secondary to and independent of synuclein. So this is a little cartoon we put together for a paper a couple of years ago, highlighting that each of these pathogenic mechanisms on their own may contribute to or cause cell death, but there's lots of basic science showing that each of these can contribute to synuclein aggregation and the development of pathological forms of synuclein that then can further drive these pathogenic mechanisms, feeding back further on synuclein and then driving cell death. So a very complex, and we don't know whether these occur in all patients, in only some patients, and what order do they occur, and what how do they combine to contribute to the pathogenesis. So pathogenic heterogeneity, we have to ask ourselves, are selected pathogenic mechanisms differentially active in different stages? For example, does inflammation only develop later, or is it an early manifestation? Does synuclein start the process and then drive all of those other mechanisms that then take off on their own? We just don't know that. And obviously, understanding this has a big impact on when you might consider initiating a particular disease-modifying therapy, or choosing one, or designing a trial with different outcome measures. Let's return to the clinical heterogeneity that I've described, and let's try to figure out how that might account for disease, and how might we explain this heterogeneity. Well, this first slide deals with whether there is the same pathology or pathogenesis, and how does that account for, or how do you fit it with the tremendous heterogeneity that I've already told you? Well, maybe that same pathology and pathogenesis initiates or develops in different locations in the brain, maybe due to differing stochastic events in different networks of brain cells that then drive the process in a different way. The same pathology and pathogenesis obviously would occur in different hosts. So I already introduced the idea of a specific HLA phenotype or genotype that then could account for differing predisposing or protective factors. The same pathology could have a different impact on different brain areas. For example, maybe dopamine cells respond to degeneration in a different way. And that's actually very logical. When dopamine deficiency occurs, you see increased dopamine turnover, increased oxidative stress, more free radical formation, and then more damage to mitochondria and other factors. So maybe dopamine cell damage has a different impact, or the pathology has a different impact on progression in different areas. Then look at the opposite side of the coin, different pathogenic mechanisms rather than the same. So maybe different pathogenic mechanisms are secondary to different variants of synuclein. This concept, different conformational variants of a pathogenic protein, is very hot nowadays in neurodegeneration. And this we've learned from the situation in Creutzfeldt-Jakob disease. I mentioned the tremendous variability in Creutzfeldt-Jakob, and we now know that in part this relates to different strains of the prion protein. And different conformational variants of the protein in synuclein could account for differing clinical phenotypes in patients with Parkinson's disease. This has really revolutionized the field. We are, I believe, on the verge of a remarkable time in neurodegenerative diseases using these seeding assays, the PMCA, protein misfolding cyclic amplification, and real-time quaking-induced conversion. Many of you might be aware of the fact we are now routinely using RT-QuIC to diagnose Creutzfeldt-Jakob disease using cerebrospinal fluid. You may not know that these same techniques are now being used in neurodegenerative diseases, and these are two hot off the press papers just this year showing that these techniques are capable of differentiating the presence of pathologic synuclein in samples of patients with Parkinson's disease. And I believe we may be on the verge of diagnostic tests for Parkinson's using such things as skin biopsies using these techniques. What about different pathogenic mechanisms due to the influence of other proteins? I've already told you about the presence of these other proteins in brains of patients with Parkinson's. Is it possible that these proteins influence the behavior of the disease. And that's actually quite logical. This is beautiful basic science work from Virginia Lee and John Trojanowski, again from the University of Pennsylvania. And what they did was they used a transgenic Alzheimer's mouse, the FAD mouse, and injected those mice with preformed fibrils of alpha-synuclein. Virginia Lee is the first to have used this, this model of injecting fibrils of synuclein and showing that you get a spread of pathologic synuclein in animals. And the mouse injection models have become a very important way of studying the disease as well as treatments. And when Virginia and her team injected alpha-synuclein preformed fibrils into this Alzheimer's mouse, in contrast to the wild type mouse at 1.5, 4.5, and 9 months, there was a profound difference in the acceleration of the synuclein pathology, as well as the presence of hyperphosphorylated tau, as you see in Alzheimer's disease, as well as neuronal loss with concomitant clinical features of cognitive decline and motor disturbances. So just this combination of Alzheimer and Parkinson proteins provides some explanation for pathologic and clinical heterogeneity. What about other diseases and other concurrent factors? Well, we know that these degenerative diseases of the brain have the greatest risk factor as aging, but also apart from the role of aging, other factors like concurrent vascular disease, amyloid, and others certainly can play a role in the way our patients present, and we need to be aware of that. And then perhaps the combination of all of the 6 that I've summarized for you. So Parkinson's, in quotes, are we dealing with a single or multiple diseases? And I'm now just going to separate this discussion from the monogenetic forms, which we do recognize must be multiple different diseases. Well, if it's Parkinson's disease, could we be dealing with a single disease with multiple modifiers that I've tried to summarize for you through the course of the last few slides with aging, post-genetics, epigenetics, strains, etc.? Or are we dealing with Parkinson's diseases, pathogenically distinct diseases? And obviously, this is a very critical question that we just simply don't have the answers for at this point. And the implications for treatment are immense. You've all seen slides like this summarizing the importance of precision medicine, and all fields of medicine are working in this area, trying to take the idea that a single treatment, one size fits all, trying to move that to the back and evolve away from that, recognizing that we have many different types of disorder that in some cases might have benefited from the treatment had we only been able to recognize those subcategories or subforms of the disease that could benefit from the treatment. But instead we lump them all together and we get side effects, we get no benefit, etc. So what we would like to do, and again, every field of medicine is doing this, is trying to stratify patients based on a variety of different factors, from clinical, demographic, socioeconomic, etc., through to biosample methods of separating these patients out. And this precision medicine approach will then allow us to intelligently separate the patients into categories that might benefit from different interventions. So let's think about the implications of these concepts, of implications to treatment, I should say. Are we dealing with a single final common pathway? Is synuclein going to be enough? Is synuclein-related toxicity, even though we're dealing with a multifaceted disease, synuclein seems to be pretty common to most brains that come to autopsy, although I have shown you that synuclein is not sufficient to cause the degeneration. Do we have sufficiently important combined mechanisms like inflammation or energy failure that our treatment can focus on those? Or possibly many molecular distinct phenotypes that require biomarker separation, precision medicine approaches with distinct treatments for all of them? We just, again, don't know the answers. And this is the kind of consideration you might be thinking if each of these circles in the Venn diagram is a different form of Parkinson's, are the commonalities sufficiently overlapping that single treatments or treatments directed at a sufficiently common process are going to be enough to provide some form of neuroprotection or disease modification, or is the overlap insufficient enough to allow that to happen? And are we going to have treatments that are distinct for each and every one of those subtypes? We just don't know. So you've probably heard of the 4 rights to precision medicine: the right time, the right dose of the right drug provided to the right patient. Well, we're going to conclude this discussion by questioning whether it should be the right drugs. And I strongly believe that's probably going to be necessary. In neurodegenerative diseases, Parkinson's disease is lagging behind Alzheimer's when we come to these arguments. There have been a number of very good papers arguing that we need to be thinking of this as a roadmap to managing Alzheimer's disease, with a number of projects conceptualizing the various approaches that we need to take. And again, this is not unique to Parkinson's at all, but you appreciate that in neurodegenerative diseases, we have a lot of challenges, for example, that cancer doesn't face. Let's look at this consideration of whether a single or multiple drug might be used. Let's think first on drug development. Well, trial designs. Obviously, you need to be thinking of how you're going to design your trial to look at these questions. It's interesting that the first disease-modifying therapy trial we ever did in Parkinson's looked at multiple drugs. And I'll just show you that in a sec. Multi-arm, multi-stage, the MAMS approach, and then basket trials. Let's just summarize what we mean by each of these. This is the DataTop trial: deprenyl or selegiline and antitocopherol, or I should say antitocopherol, antioxidative therapy of Parkinson's disease. A study, I couldn't believe it when I was just reviewing these slides. We published this in the New England Journal going on 30 years ago. That's how long I've been doing it with neuroprotection. We're still failing. But you just see here, it was a study of selegiline combined with tocopherol or vitamin E in a 2x2 factorial design. Interestingly, this is, to my knowledge, still the only multi-drug study that has been done in disease modification in Parkinson's. The concept of multi-arm, multi-stage trial really has been championed in cancer, but it's logical that we should be thinking about it in neurodegeneration, the idea that you can have a single control arm and then multiple treatments, some of which have to stop because of futility or adverse events, and then you can start up new treatment arms, and then by the time you're finished, you have final results of 2 or more drugs. This was championed with prostate cancer to begin with. And then the idea of using basket trials. This is an important review paper that I'd refer those that are interested, published in New England Journal a couple of years ago. And basket trials study a single targeted therapy in the context of knowing that you're dealing with multiple diseases, and in the era that I'm talking about in this lecture, multiple disease subtypes or even versions of the same disorder. So here's a single genetic mutation in multiple different types of cancers, with the idea that you can lump all of these patients, independent of the type of cancer they have, into a single trial and gather important information on outcomes. Now, basket trials are just now being studied in neurodegenerative diseases. To my knowledge, this is the first one, led by my colleague Adam Boxer at UCSF, published just in the last year, where he studied this microtubule stabilizing agent. This is an anti-tau agent. I mentioned Alzheimer's disease has tau as well as A-beta as the 2 important proteins, but progressive supranuclear palsy and cortical basal degeneration are 2 other tau diseases. And most people wouldn't have thought to lump these diseases with Alzheimer's, but it does make sense if you've got a drug that is designed to affect the pathologic form of tau. Unfortunately, the drug didn't work, but this was the first attempt at basketing neurodegenerative diseases that seemingly are very different. Second slide on this concept is now what arguments for multiple drugs as they apply to disease pathogenesis. If we're dealing with multiple distinct biological disorders or even molecular phenotypes of the same disorder, obviously multiple drugs may very well be required. It may be that we're dealing with multiple contributing biological mechanisms, even in a single disorder versus those multiple molecular phenotypes. But even if we're dealing with a single disorder, a single drug may have insufficient impact to slow the disease progression. So it may need to be added because a single common pathway may not be sufficiently impactful or strong, or maybe the synergistic effect of multiple pathogenic factors are required and overwhelm the individual effects. So again, mitochondria versus other energy factors versus inflammation, et cetera. Maybe the synergistic effects of the combination are more important than the role of a single one. So again, looking at this, you can imagine that maybe if you've only got a treatment that affects mitochondria, it's the combination of factors that are creating the lion's share of the cell death. And then finally, this idea that even if we're dealing with a single component that you're interested in, maybe multiple drugs targeting different components of a single pathway could be the way to consider treating. And this comes to alpha-synuclein. This is a cartoon from Dmitry Krank a couple of years ago. And we're now using these concepts in drug development, alpha-synuclein being the main pathogenic protein. So maybe we can alter synthesis, aggregation, increase its degradation, change its propagation and uptake into other cells, actively immunizing against the synuclein. And so recognizing the various different potential places you can direct your therapy, maybe multiple approaches combined. So you could imagine combining passive immunization, which we are doing, and with monoclonal antibodies against synuclein now. We're doing this in clinical trials. You could maybe combine that with agents that stabilize the aggregating protein or alter its synthesis. And so the idea of multiple drugs along a single pathway may be something worth considering. Let's come back to this slide that I showed you before. Parkinson's disease, distinct biological subtypes. These were the cancer forms. Now let's think of many different forms of Parkinson's. Do we need biomarker-driven trials only in selected populations? Will a drug be only useful for this second type of Parkinson's disease, but not useful for the fourth type? Alternatively, is it possible that each of these types have selected pathogenic mechanisms that they share? And maybe if that's the case, we can then think of using basket trials, combining these patients that have these overlapping pathogenic mechanisms. Targeting them may not be as effective as using a treatment for a selected biological subgroup, but still, this may be useful for providing some degree of disease modification. However, I think that as in cancer, combined therapies or cocktails still will have a greater likelihood of benefit because it may be insufficient just to get that factor. You may have to get the more common feature in this group of patients with Parkinson's. So I think this may be my last slide or close to it. Neuroprotection for Parkinson's disease. The one drug does not fit all question. Let's think, what do we mean by one drug? Are we talking about a single drug versus the same drug, but in combination with others? And again, we don't know what we're going to end up with. What about all? What do we mean by all? Do we mean all patients that we think have clinical Parkinson's disease today, where we don't have any biomarkers that allow us to separate out subtypes? Or are we talking about patients with very selected, biologically defined subtypes that we can't characterize just yet? And so let's stop at that point. I hope that I've intrigued you a little bit. I hope that I've shown you how complex the field is. I welcome all your questions and comments, and I'll stop at that point. Thanks for your attention.
Thank you, Tony. That was amazing. The level of complexity of Parkinson's disease and its treatment really inspires me to stay in type 1 diabetes after all Until it makes the blood sugar go down.
That's an easy disease. Come on.
Right, so it's fairly. So I'd like to open this up to questions from the audience. Please use the chat function on Zoom, or alternatively, you can raise your hand and ask a voice question. If you could raise your hand first, though, because we have a lot of people on, or use the Twitter tag, and that'll go directly to Christian. There's a bunch of comments thanking you for your presentation. Opening it to questions now, please.
There are some questions in the chat.
Okay, let's see. Oh, wow, this suddenly appeared. Okay.
I'll stop sharing my screen, maybe.
Yeah, so if alpha-synuclein is a functioning protein, how does targeting it, say by antibodies, avoid dysfunction?
Great question. We know, in fact, that in animal models, you can knock out synuclein and still have a very functional animal. So we know that at least reducing synuclein, patients, we hope, will function well without consequences. However, it's an excellent question. And in all of these diseases, we have to consider gain of function versus loss of function models or questions. Most of the treatment we think of as gain of function, with the toxic effects of synuclein being preeminent. But synuclein, as I mentioned, is a synaptic protein that has a great deal to do with release of transmitters, reuptake and vesicularization of transmitters at the terminals. And so it's logical that maybe loss of function does contribute somewhat. So it is an important question.
Thank you. Just out of curiosity, though, in terms of using antibodies, are they having to be taken up into nerve endings? I mean, antibodies are fairly large proteins. Are there transport processes that allow that?
Yeah, so both active and passive immunization is now being studied in Parkinson's. The first large-scale passive immunization study just reported its 52-week outcome. Unfortunately, primary outcome negative, but secondary outcomes really very interestingly positive so far.
Great.
And we're just about due for the 52-week Biogen results. We expect that these antibodies have to cross the blood-brain barrier, and they do so minimally. So only about 0.3% of the infused antibody actually gets into the brain. However, those are in levels that, at least in animal models, are believed to have sufficient impact on the extracellular synuclein. So it's not the synaptic synuclein it's influencing. It's I didn't get into any of the discussion of cell-to-cell transmission in neurodegenerative diseases, but it's believed that the synuclein has the capability of being released in its pathologic form by one cell and being taken up by neighboring cells, and then by its method in a prion-like fashion, by what's called permissive templating, it induces the abnormal folding of the normal protein in the cell that's taken up the pathogenic protein. So we're trying to take up the extracellular protein before it's taken up by the next cell.
Great, thank you. A question from Jillian. Has the microbiome been implicated in Parkinson's disease?
Boy, that's a very hot topic that we could spend at least another hour on. Yes, it's been implicated. There's a lot of information in the literature. I've been collaborating very actively with colleagues in Malaysia who are doing some phenomenal work in the microbiome. And it's in fact, probably not just the microbiome, but the micrometabolome, the microbiome's metabolome, and all the metabolomics of those bacteria in your gut that probably have an important role to play. And so the concept of the microbiome is very naive, because you can change your diet or change where you live and your microbiome changes immediately. Constipation causes a change in microbiome, and constipation is common in Parkinson's disease. So the interpretation of the just flat microbiome is probably not where we need to go. We need to think about much more complex interactions.
Don Redelmeyer congratulates you and asks about prevention of Parkinson's.
Prevention in Parkinson's. Well, a whole field in Parkinson's is taking patients that we believe, or individuals at least, that we believe are in a prodromal phase of Parkinson's. Now, the obvious subgroups that will probably be treated in the earliest types of studies with this in mind are those that have mutations of the known genes. So if you have a LRRK2 mutation, or certainly an alpha-synuclein mutation, those individuals will almost certainly be in preventative therapies in the next little while. It's important to acknowledge, however, for example, in LRRK2, that the commonest mutation in LRRK2, the so-called G2019S mutation, is only about 30% penetrant. And we still don't know what those mechanisms that make it penetrant or prevent those that don't eventually get the disease. We don't know what those mechanisms are. We have some preliminary studies and some that we've been contributing to here in Toronto, but there's a lot more work that needs to be done if you're going to put a whole slew of people with LRRK2 mutations into a preventative trial. But there are a lot of studies on this field. I'll just mention one really interesting area is, you may not know, but a condition called rapid eye movement behavior disorder, or REM behavior disorder, is a condition where you act out your dreams. You have dream enactment because your brain has lost the ability to shut off motor control in dreams. So we have a normal brain mechanism that makes you atonic to protect you from bashing your wife in the face, falling out of bed, injuring yourself. And believe it or not, in Parkinson's disease, up to 20 or more years before you develop Parkinson's, you lose that normal REM atonia. And so you act out your dreams. And so that is one of the earliest features of a synucleinopathy. And there's a lot of interest in enrolling people with what is called idiopathic REM behavior disorder in preventative trials.
Interesting. Let's see, other questions. Neuroepidemiologically, pesticide use has been implicated in Parkinson's disease. Is this still the case? And any ideas how it might relate?
Yeah, so it is definitely the case. You need large epidemiological studies to show this. So obviously lots of people have been exposed to pesticides that don't develop Parkinson's, and vice versa. Many people with Parkinson's are never exposed. But if you take the large epidemiological studies, certainly herbicides and pesticides do seem to play a role. It is likely that these can be toxic to mitochondria and to other factors that were on that slide where I showed you the various different components of Parkinson's disease. So we do believe they can play a role. It may very well be, you know the old adage that, what is it? Genes load the gun, environment pulls the trigger. And so in fact, it is likely that your genetic makeup is actually quite critical to your response to the pesticides, for example.
Great. Thank you. Any final questions for Dr. Lang?
Tony, this is Dan Drucker. Fantastic lecture. Could you say a word about the possible neuroprotective therapeutic potential of the GLP-1 receptor agonist class, and where you think that might go or not go?
Yeah, it's certainly one of the classes of agents that look very promising for a variety of reasons. As you know, these agents have trophic effects, anti-apoptotic effects, a variety of other pro-mitochondrial effects. There are studies that have been undertaken, some with promising effects and others somewhat disappointing, but it may be that the more selective agents and those that get into the brain more effectively will have greater impact, and they certainly are being very actively followed up. And in that one of those slides that I showed earlier on, I think there are probably about 4 or 5 GLP-1 receptor antagonists that are being studied.
Another question, have there been any advances in terms of understanding PD given the evolution of AI?
the evolution of patients or humans or AI.
AI.
Listen, it's actually a very interesting question because it has been regularly shown that Parkinson's disease doesn't occur in any lower species. And so, in fact, evolution may very well play an important role, and even non-human primates, although we can induce Parkinson-like pathology by putting synuclein into brains, abnormal synuclein into brains. But in fact, spontaneously, Parkinson's disease doesn't occur even in old world monkeys. AI is significantly contributing to search for drugs. One of the areas that we've used the IBM Watson in is to find agents that might have disease modifying effects. Lorraine Callia in our group has used Watson to look for agents that interact at a cellular level with synuclein. And she's then been exploring some of these agents in her cell models of alpha-synuclein. So yes, AI, I think, is being actively used to pursue some of these issues.
Another question from Lisa Richardson.
Sorry. Hi, Tony. Thank you. That was amazing. I'm sitting behind my grandfather Rick's desk right now at Toronto General Hospital, and actually just thinking about like what he would be thinking if we were to see this field now. It's just unbelievable, the incredible work that you've done. It makes me think about when I think about precision medicine, and I actually think about Rick and think about his obsession with the clinical exam and the physical and clinical evaluation. How do we balance precision medicine with the other important aspects of personalized medicine, like the clinical exam, the patient-physician rapport, and all of those other factors? How do we not lose track of that?
That's really a great issue, and it's kind of interesting considering the era we're in right now, how we have to ask these same questions in terms of our virtual contact with patients versus our live hands-on connection with patients. I should also mention it's just an absolute privilege to be asked to give a Levinson lecture, someone that I work with at a senior level in medicine, and now be referring to Rick Richardson, who I followed as the head of neurology, and I gave the first Richardson lecture in neurology as well. So it's a real privilege. At least at present, you can't make a diagnosis without clinical. The diagnosis remains clinical. I think despite all of the new approaches that are going to be really critical in confirming the presence of synuclein pathology, it's still going to be very important to put those into perspective with the clinical consequences of those changes. And so I think we're going to be examining and taking care of patients for a long time to come. And even if we don't need the clinician to make the diagnosis, you still need the clinician to hold the hand of a patient that's got a bad degenerative disease. And until we have effective disease modifying therapies, boy, that's really critical. And these are all tough diseases. And the disease that your father's name is linked to, Richardson syndrome, a progressive supranuclear palsy that your grandfather's linked to, is much, much worse than Parkinson's, another proteinopathy with tau. And so we're going to need clinicians doing a good job for a long time to come.
So one last question. This is from Dr. Nakor. Great lecture. Thank you. How soon are we using RT-QIC for diagnosis here?
Well, it's amazing. With COVID, our biomarker program that is led by a new recruit to our hospital, and all of you should know this name, Gabor Kovacs. We recruited, he's a neurologist, neuropathologist. We recruited him from Vienna. He now leads the basic science program for our progressive supranuclear palsy Rossy program. And his laboratory has this technique nailed in a way that we believe, but still needs to be proved, may be better than most other centers. We have preliminary evidence that it may allow us to distinguish Parkinson's from another synucleinopathy, multiple system atrophy. And COVID has held us up in getting institutional approval to do a biomarker study looking at CSF, skin biopsy, blood, saliva, and sebum. And so beginning, we hope, next week, we will initiate that project. We're doing it in a large number of brains that we now house in our brain bank. And we hope by the end of the year, we're going to be publishing some really, really important data that then will be the precedent to being allowing us to use this technique. So I think literally, I've already been contacted by one company that says that it wants to start doing RT-QuIC of alpha-synuclein in the cerebrospinal fluid for diagnosis. So we're there very, very soon.
Wonderful. So on that note, I know that there are other questions. If you could contact Dr. Lang offline.
I'm glad to answer any questions if you want to contact me.
But
UHN Research.
Yeah, so I just want to thank you again, Tony. This was an absolutely marvelous talk. And Wendy, we hope that you felt honored that we could have such a great presentation for your lecture. Anyway, so everyone, thank you very much for attending. Please don't forget to fill out the online form. You can also get credit for this as well. There's a link on our website as well as a SurveyMonkey link. So please fill it out. And again, thank you very much, Tony, and everyone.
Thanks, everybody.
Bye. Stay safe.
Bye.
Take care.
Stay safe, everyone.
Wonderful talk, Tony. Thank you.
Thanks, Arno. Take care.